So we've unzipped it, we've copied it. Okay. This concept of taking this genetic material from inside our nucleus, copying it, getting it out into the cytoplasm, translating what we've copied down, and make stuff is referred to as the central dogma of biology, really. It is the central dogma of biology is right here. The flow of the genetic material, the DNA, okay? Getting that copied, creating what's called RNA. Taking that RNA, getting it out into the cytoplasm and translating it and making a protein. And then that protein can go and do whatever it's supposed to do. That is the central dogma of biology. It's one of the bigger concepts of biology. We got, this as a major, concept in biology. Evolution is a major concept of biology. The concept of cell division, the cell theory. Cells come from pre-existing cells. What makes up a cell? These are some of the kind of the bigger major concepts that we have in biology here. So let's look at what's called the genetic code of life here. All right. It is, again, DNA, right? But we have genes within that DNA that really specify information. Okay. It is the blueprint, if you will, for what we need to make or what is the function that needs to be accomplished. Okay. When genetic information is expressed, it's made into structures. If it's expressed, what in the world? Okay. It is made. And the structure will dictate the function. And the function is dictated through that genetic code. Okay. Getting the information out of DNA is dependent upon RNA, ribonucleic acid. Okay. Ribonucleic acid is ribose is the sugar and not deoxyribose. It's single-stranded, not double-stranded. It has 4 nucleotides, adenine, guanine, cytosine, and uracil, most commonly screwed up thing on the exam. Okay. No thymine, uracil. Okay. But adenine and uracil, similar structure like adenine and thymine, so adenine always binds with uracil. Okay. Uracil, thymine, uracil. Okay. So DNA, double helix, deoxyribose sugar, thymine as one of the nucleotides. RNA, single-stranded, ribose is the sugar, and uracil replaces thymine. Okay. When we look at RNA, it helps us to get that information out of the DNA, get it out into the cytoplasm so we can actually do some stuff with it and copy some things down. We have 3 types of messenger RNA. Three types of RNA. mRNA, messenger RNA, ribosomal RNA, rRNA, and what's called transfer RNA, tRNA. These are my 3 types, and I would know the difference between these types of RNA. Messenger RNA, we're going to see this one first. It takes the message from DNA, again, in the nucleus, okay? And this takes it to the cytoplasm. So this is translating it, okay? Or I'm sorry, transcribing it. It is making a copy of it inside the nucleus. So mRNA is there in the nucleus. It's copying everything down, okay? It's like, okay, I'm going to take this information out into the cytoplasm, okay? I'm going to write everything down. That's what messenger RNA does. It leaves through the nuclear pore, goes out into the cytoplasm, and there it's going to interact with the other 2. Ribosomal RNA is what makes up the ribosomal proteins. Okay, Remember ribosomes from way back when, right? Ribosomes, they said there were 2 ribosomal subunits that make up a ribosome. Ribosomal RNA helps to comprise these 2 subunits to make a ribosome structure. Transfer RNA helps to transfer, see what we did there? The right amino acid to the ribosome. So tRNA is going to be involved with the actual translating of this thing, where mRNA and RNA are interacting with one another, reading it, and saying, hey, yo, tRNA, get over here. Bring that amino acid. And tRNA brings the amino acid to the party, elongates, makes the protein. Ta-da. So what's in this genetic code? It is letters. It is uracil, adenine, guanine, and cytosine. Okay. These are referred to as codons. Okay. And each of our 20 different amino acids, okay, you have 20 amino acids, 9 are essential and 11 are not essential. Okay. Essential means I have to go get them in my diet. You can make 11 of them. You can't make 9 of them. So, but there are 20 in all. These letters create the alphabet. Can't make an arrow today. Create the alphabet that is referred to as codons. Okay. So what does this look like? Well, here you go. Here are all my messenger RNA codons. And in no way I'm going to ask you what all of these are. Okay. I mean, if you go further on into biochemistry and whatnot, you'll probably go into some of these. But this is the genetic code that all of our bases are going to be derived from. Okay. It's these 3 letters. Okay. Just 3 little letters. These are the words of genetic information. It's the shorthand. Okay. Messenger RNA is just making a shorthand version of the directions found in the nucleus in the DNA. Okay. I'll kind of 64 arrangements. Okay, there's 64 words. There are, as you can kind of see, 3 stops. And only 1 start. Okay, only 1. So when we're translating the sucker, you can have an really long copy of it, but messenger RNA is only going to start translating where I have the start codon, AUG. It finds AUG, and that's where it begins. Okay. There could be a few codons in front of that. Okay. But it doesn't matter. There could be a bunch of letters in front of that. It does not matter. It can only start at the start codon. There's only 1. Only 1. Multiple stops, only 1 start. Through just these just through these 64 different words, okay, we can make a multitude of various different things. Everything that you see on you that's protein related, which is pretty much everything, all of that comes from uracil, cytosine, adenine, or guanine. Okay. U goes first, or a C goes first, or an A goes first, or a G goes first. So there is a multitude of various different outcomes that these words can give, just like the English language, right? Several different letters come together to give us a plethora of various different words. So with that said here, the genetic code has 4 basic properties. It is universal, okay? It is universal. All organisms use the same words in the same way, the same code, okay? There's exceptions to every rule, but all of them, from all intents and purposes, okay, all organisms will use this code the same way. They encode for the same 20 different amino acids with the same 64 triplets. So this is universal. It is redundant, okay? There are 64 codons available for 20 different amino acids. So most amino acids are encoded by 2 or more codons. So that's redundant. We have multiple words to code for the same thing. They are exclusive or unambiguous, okay? Each codon specifies only one of the 20, okay? So I can't have a 3-letter codon code for either this or that. It's got to be one or the other. So it is exclusive. It is non-ambiguous. If it was ambiguous, it could be either or, okay? Flippy floppy nonsense. It is unambiguous. It is exclusive, okay? AUG is always methionine and is always a start codon. It doesn't code for anything else. And again, we have a start and we have a stop. We have a capital letter, AUG methionine, and then we have the 3 stop codons, all right? So these are properties that that genetic code shares across all life here, okay? Universal, everybody shares it, all coding for the same nonsense. It is redundant. Some codons will encode for 2 or some amino acids will be encoded by 2 or more codons, 2 or more words. It is exclusive. Each codon only does this. AUG only is methionine, okay? ACG is always threonine, all right? It is redundant. ACG is not threonine, or I'm sorry, lysine. It is always threonine. And there's a start and there's a stop, okay? Probably something important to remember.